I'm going to begin my talk with you today by talking about a project that I've been working on for the last 13 years, almost exclusively, a work entitled the Xenotext. Now, my presentation is sufficiently pretentious enough to have two epigraphs. The first is from F.T. Marinetti from the Technical Manifesto of Futurist Literature, where he says that there is also a microbe essential to the vitality of art. And I'm going to talk to you about such a microbe. I've also included this quote by Prince Philip of England, who notes that in the event that I am reincarnated, I would like to return as a deadly virus. Because he would like to solve the problem of overpopulation. The work I have in mind for you is a work of genetic engineering. The Xenotext is a kind of experiment, a literary exercise that explores the aesthetic potential of genetics in the modern milieu, doing so in order to make literal the renowned aphorism of William S. Burroughs, who has declared that the word is now a virus. My experiment proposes to address some of the sociological implications of biotechnology by manufacturing what I have called a Xenotext, a beautiful, anomalous poem whose alien words might subsist like a harmless parasite inside the cell of another life form. I've composed my own example of living poetry, so that when translated into a gene and then integrated into the cell, the text gets expressed by the organism, which in response to this grafted genetic sequence actually begins to manufacture a viable, benign protein, one that, according to the original chemical alphabet, is itself yet another text. I am, in effect, engineering a primitive bacterium so that it becomes not only a durable archive for storing a poem, but also an operant machine for writing a poem in response. Here is the host for this project. This is the extremophile bacterium Deinococcus radiodurans. It is the proposed symbiote for my Xenotext, in part because this extremophile can actually repair its own DNA so quickly that the germ resists mutation. The organism is a kind of evolutionary dead end. It does not evolve. It repairs its DNA so quickly that it does not mutate. But it is so well adapted to the lethality of the universe that it does not need to evolve. Scorch it, freeze it, wither it, and still it endures. It can survive exposure to the vacuum of outer space. It can withstand dosages of gamma rays 1,000 times more lethal than the dosage needed to obliterate a human being. A few biologists, including Anatoly K. Pavlov and others, have even gone so far as to argue that the ancestor of this organism must be extraterrestrial in origin, that this organism must have spent at least part of its evolutionary history in outer space in order to have acquired all of these environmental immunities. Consequently, it is a very durable archive for storing a poem. I'm about to show you the actual works stored in such an organism. Because there exists a codependent biochemical relationship between any preliminary genetic sequence and its resulting protein sequence, my two poems must likewise be bijective in their structure. So I try to explain this project by telling people, imagine pairing off all the letters of the alphabet so that they are mutually assigned to each other. So if, for example, I wanted to assign A to T, I have to assign T to A. If I assign N to H, I have to assign H to N, etc. Let us imagine pairing off all the letters of the alphabet so they're mutually correlated, knowing that there exist 7,905,853,580,625 possibilities for such encipherments. Just a little shy of 8 trillion ways of enciphering the alphabet according to this constraint. Now pick one of those ciphers, pick one of those 8 trillion possibilities, and write an eloquent poem such that if we replace every single letter with its counterpart from this code, we get yet another eloquent poem. It took me 4 years to write these two poems. this constraint turned out to be far more burdensome than I ever imagined. After nearly 4 years of failure, trying to write 2 intelligible texts that were meaningful and had poetic merit, according to this very burdensome constraint, I have nevertheless made the prerequisite breakthrough, and here are the 2 poems. Okay, the poem that you see here on the left is the one that I have written and enciphered as a DNA sequence. It's a poem that I have nicknamed Orpheus. The poem on the right is the poem that the organism creates in response to my text. It reads my poem, and in response it builds a protein which enciphers the text you see here on the right. And I've nicknamed this poem Eurydice. Now, in part, I've nicknamed these 2 poems with these mythological names, in part because of the somewhat demonic, infernal turn that this project has taken. It's taken on a kind of hellish overtone. So I'm going to read to you now the poem on the left, the poem that I've called Orpheus. And it begins, "Any style of life is prim. Oh, stay, my lyre, with wily ploys moan the riff of any tune aloud, moan now my fate. In fate we rely. My myth now is the word of life." Now the organism actually reads this poem, and in response it writes the poem you see here on the right, a poem I've nicknamed Eurydice, and it reads, "The fairy is rosy of glow, in fate we rely. Moan more grief with any loss is the achy trick, with him we stay. Oh, stay, my lyre, we wean him of any milk is rosy." Now, when I point out these 2 poems to most people, they say these aren't the kinds of poems that are typical of Christian Bök. However, they suddenly have an uncanny response when they notice, for example, that wherever you see an A in the text on the left, you'll see a T in the text on the right, and vice versa. Wherever you see an N in the text on the left, you'll see an H in the one on the right, and vice versa. The 2 texts are perfectly transposable all the way through the entire poem. The text on the left is written by me as a kind of masculine assertion about the aesthetic creation of life, while the text on the right is written by the microbe as a kind of feminine refutation about the woebegone absence of life. The 2 poems are deliberately in dialogue with each other, and in this respect the 2 poems resemble Petrarchan sonnets, they're 14 lines, in dialogue with each other, much like poems written in the elegiac pastoral tradition of the herd boy addressing the nymphette. Moreover, here's the really cool part, the protein that enciphers the poem by the microbe has actually been chemically tagged so as to make the cell glow red in the dark. When the organism is writing its poem, that poem causes the organism to fluoresce and turn red. The microbe is in effect able to fluoresce rubescently in a fey way that actually embodies the rosiness attributed to the fairy described within the content of the poem itself. I am in effect trying to transform this extremophile organism into something akin to a little fairy that glows red. And in fact, that is how you recognize that the organism is doing its job. It begins to glow. On the 31st of March in 2011, my lab at the University of Calgary confirmed that my gene XP13 caused a culture of E. coli to fluoresce red in our test runs, implying that when implanted into the genome of this bacterium, my poem caused the cell to write its own poem in response. And I enjoyed about 6 weeks of international renown for having reached this benchmark. However, later tests showed that the protein was only half as massive as expected, implying that the germ was either censoring my poem or destroying it during expression. I had in effect engineered not the first microbial writer, but the first microbial critic. This was not good news. I had to go back and try again. The scientists wanted me to prove that I could actually engineer a colony of E. coli, a colony much more easy to modify genetically, and less expensive than the extremophile. So I had to prove the concept first and then transplant that construct into the more expensive and more difficult organism to engineer. So getting to work in E. coli is an important step. It took me 5 failed assays to figure out how to get the protein to be viable enough and stable enough to function properly in the organism. On October 3rd, 2012, after months of many failed assays and a lot of money spent, I finally received confirmation from the lab at DNA 2.0 that my revised genetic cipher gene XP13-4A did cause its culture of E. coli to fluoresce properly without destroying the poetic matter of the protein. So now I can say that I am the first person in history to design a microorganism capable of writing a meaningful text in response to an enciphered gene. Now, I've since collaborated with a lab at the University of Wyoming in order to implant this gene into Deinococcus radiodurans. And I should note that that dark blot in the second lane from the left of the slide, that signals the presence of the poem in E. coli. That is, in fact, the poem represented by the protein, manifested there detectably in its full mass. So this is an important image to me. To have seen this image materialize with that blot where it was supposed to appear was good news. Now, if you're curious what this protein would actually look like, at an atomic level, this is the computer simulation of the way this protein folds. This image is the backbone of the resulting protein after 2 femtoseconds of folding, according to computer simulation. It's a relatively good textbook case of a protein with numerous alpha helices coiled up like spaghetti in a very compact formation. If you'd like to know what that looks like atomically, here is the actual atomic structure of that protein transposed over its spinal structure. This image depicts what the poem is expected to look like at the atomic level in the cell. Now, of course, this is in effect a kind of atomic translation of the work. I'll show you this image, which depicts the molecular embodiment of the Xenotext as a kind of sculptural translation of the poem itself, a model actually made out of toy molecules for exhibit in a gallery. And here you see it at the power plant in Toronto as part of a major retrospective exhibition on conceptual literature. But this protein has appeared at numerous other galleries around the world. On October 31st, 2013, after months of detailed planning, I received word from my lab at the University of Wyoming that the implantation of the Xenotext into Deinococcus radiodurans had achieved 2 of my 3 benchmarks for success. First, the organism had fully integrated the plasmid for the gene into its chromosome, meaning that the gene would actually endure in the cell. This was considered a very difficult step, and the scientists were blown away that an English major like me actually produced a viable genetic sequence that could function in such an organism. Moreover, the organism was in fact fluorescing red in response to the gene, meaning that the cell was constructing the protein according to our expectations. The technicians, however, could not detect the full mass of the protein, arguing that the organism was metabolizing the construct too quickly for their methods of detection. So again, I had the same problem I encountered with E. coli initially. I basically took the successful candidate, a structure from E. coli, and transplanted it into this new organism to see what would happen. And alas, from my perspective, it failed. Nevertheless, the scientists thought that they were conveying good news about my success when, of course, these results implied that, as in the case of failed assays for E. coli, the extremophile was destroying the poetic matter of the text before we could in fact read it, meaning that no one could, of course, verify the poetic merits of the microbe. Effectively, I was sending a fax straight to the shredder before you ever had a chance to read it. Now, this picture, of course, depicts the failed sample culture of my synthetic bacterium. This is the offending organism right here, the culture that did not work. Alas, I found out this result on the very last day of my funding. I actually got this answer on the very day when all of my contracts expired and all of the money I had at my disposal was finally spent, when all the budgetary constraints had been used up. I had spent more than $120,000 Canadian over the course of 5 years on this project. Nobody gets $120,000 advance for a book of poetry, right? I had spent $120,000 over 5 years, only to fail after a photo finish, stalling out within sight of my finish lines, bereft of any hypothesis about the reasons for this failure. Consequently, I've now set out to raise more money to rebuild my team so that I can resolve these issues in the hope of actually making the poem work. I'm too close to having succeeded after nearly 13 years of work. Now, had I simply promised to create a microbe with the capacity both to store a poem and to write a poem, as was the case definitively for my successful experiment with E. coli, I would undoubtedly be among the most famous living poets of the 21st century. But alas, I have in my hubris promised too much, exceeding the ambition of putting a chimp into orbit so that I can instead put a chimp on the moon. I have learned that if you promise to put a chimp on the moon, nobody cares if you actually put the chimp into orbit. I have been working hard to ensure that as an act of conceptual literature, this project does not exist merely in thought alone. And here, of course, is the excerpt from The Alien that really, I think, summarized my attitudes towards the failure of this experiment. This is how I felt at the end of the whole process. It was priority one, ensure return of organism for analysis. Everybody else, all of you, you are expendable to me. All right. The Xenotext ultimately infects the language of genetics with the poetic vectors of its own discourse, doing so in order to show that through the use of such biological emissaries, we might transmit messages across stellar distances or even epochal intervals. Unlike any other cultural artifacts so far produced, except perhaps for the Pioneer probes or the Voyager probes, my poem, stored inside the genome of a bacterium like Deinococcus radiodurans, might testify to our presence upon the planet until the very hour when at last the sun itself explodes. I am in effect trying to write an immortal book, an artifact that can survive our own extinction. Now, even now, a copy of the poem has hitched a ride, not only aboard the OSIRIS-REx probe en route to the asteroid Bennu, and here you actually see the certificate verifying that yes, my poem has actually been included among the payload of this probe, but it is also aboard the MAVEN probe en route to the planet Mars. Moreover, a radio telescope at the Jamesburg Earth Station in Carmel, California has beamed a copy of the Xenotext to the exoplanet Gliese 526 in the constellation Boötes as part of a plan to communicate with any civilization that might reside on this allegedly habitable world orbiting within the Goldilocks zone of its red dwarf star. As you can see, I'm in effect striving to create an imaginary solution to many of the requisite problems that poetry faces on the eve of its own demise, a demise already upstaged in advance by other cultural concerns more epic than any poem.